Okay, well, welcome back. We are now starting up part two of day three of the 2025 ISSRD conference technical sessions. We're very glad to have everybody here, and we have a really good set of presentations coming up right now on human health and performance. I'm not going to waste any time with more introductions or anything like that. So let me just turn it over directly to Narasimha Prasad, who is at NASA Langley Research Center. Prasad, take it away.
Thank you so much, Jim. Good morning, everybody. My name is Narasimha Prasad. I'm an aerospace technologist as well as the advanced material leader for the in-space production applications. And I will be your chair for this session. Here is how we will proceed for these virtual technical sessions. And to start with, we will present the first 5-minute executive summary, and we will then open up for questions for that author. And if you have a chance, could you please enter your questions in the question and answer section? And we'll then move on to the next presentation and author and so on and so forth. And after each presentation, we will have that author come on screen and begin the question and answer from anyone here in this webinar. And webinar attendees will not be able to come on screen. So please use the question and answer tool to submit any questions you might have. If you have time left at the end of the session, we can address any additional questions that might come along. And also you could tag along with the author after the session is done. And by the way, if you want to see the full 20-minute video presentations, please visit the online program and open up that file. Now let's start with the first presentation. Jim, go ahead.
I am Rahul Goel. I am the lead scientist for human research and health applications at Axiom Space. Today I'm going to provide an overview of my presentation, human health and performance research on Axiom Space's private astronaut missions to low Earth orbit. So if you look at this presentation, I will be talking about, first of all, our mission and objectives. Our mission is simple, which is to make space accessible to everybody. And towards that mission, we are working towards three main objectives, which is building a commercial space station to replace the ISS. We are creating the next generation of spacesuit to be used on the moon and the Axiom station. And we are partnering with countries and private astronauts for missions to the ISS and eventually to our future station. In this presentation, I'm going to talk a little bit more about our background for people who may not be familiar with. I'll talk about a little bit about our plans for the station, the roadmap for the station. I'll talk a little bit about the LEO suits and the lunar and the LEO suits that we are developing, and then I will get into the private astronaut missions, the 4 missions that we have done, the type of research we have done, the 4 missions, Axiom 1, Axiom 2, Axiom 3, Axiom 4. Then I'll go into a little bit of detail about some of the projects, some of the human research projects from all of these missions. I won't go into detail for all of those projects. A lot of that information is on our website, but I will highlight a few of those projects just to show the variety of projects that we have supported across all these missions. After that, like I'll talk about a particular study from Axiom 4, which was called Sweet Ride, and it was to enable eventually insulin-dependent diabetic to fly to space. We did some tech demo to do some baby steps required to be able to support such a mission. I'll talk a bit about research in numbers. So far, we have supported more than 160 payloads across our 4 missions, and a majority of them have been human research and life science as well as the second biggest category. So far, across all those missions, we have had 15 peer-reviewed publications. They are mostly from Axiom 1 and 2 because it's not much time has passed since Axiom 3. At least from the first 3 missions that I'm aware, we have had 50 conference abstracts as well. And these numbers are only from our PAM flights, the private astronaut missions. We also have supported nearly 40 experiments so far on CRS missions in the last 4 or 5 years. After that, I will talk about some selected publications from our human research experiments from the first two missions. There's this peer-reviewed book from Dr. Shera from Saudi Arabia. It talks about a couple of neuroscience research projects, some new methods that were used for the first time, like functional near-infrared spectroscopy, and some details about that. We'll talk about this study from Trish, which have been our partners right from the very first mission. They are trying to build upon this commercial astronaut data repository and are doing an essential omics and essential measures across all these studies. I'll talk about a little bit about this study from Axiom 1 mission on changes in ocular rigidity and choroidal thickness in response to microgravity. The study on plasma biomarkers from the first all-civilian mission to the ISS. Some studies looking into pain responses. Then I will talk a little bit about potential areas of collaboration that we are very interested in, things like medical data architecture, any technologies to carry out blood analytics without refrigeration, etc., point of care ultrasound imaging, packing solutions for medicines to facilitate operational use, novel exercise capabilities, new technologies to support behavioral health, technologies to test enhancing shelf life and palatability of food, better understand radiation, again on exercise, what is the minimum bound of exercise that we can have so as to maintain reasonable fitness for the astronauts. And with that, I'll conclude. And if you have more questions, you feel free to reach out at research@axiomspace.com. Thank you.
Oh, thank you, Rahul. It's an interesting summary of what Axiom is doing. And I'm looking forward for some questions from the audience. Before those questions arrive, I have a question here. What is your immediate and prominent mission coming up soon?
Yeah, so we have just concluded our fourth private astronaut mission in July. So we are waiting to see. We have applied for the PAM solicitation. We are waiting to see the next mission if we are allotted that. But we're hoping that the next 2 years or so we will probably get one more mission to go to the ISS, or once we have our station modules, free flyers. In maybe 3 years from now, then we are hoping to get more missions lined up at that time. I do see a question.
Question,
Uh,
yeah.
should I take that question,
Yeah,
or you
you
may?
please take the question. Yes.
Yeah, I saw one question. I was like, it was just like a teaser for the bigger presentation. So
Yeah.
I was probably the first one to upload it. I didn't know like how this short one has to work. So I used the same slides. I just went through them quickly, saying that what is there in the bigger presentation. So sorry about that if you didn't get much chance, but encourage you to go and look at the longer presentation that may have more details. But it's the same number of slides in the longer
Okay.
presentation. Yeah.
Also, there is another question. Are there any opportunities for students, grad students, to intern with your Axiom space?
Potentially for next summer, it's little too early to have those positions open right now. I would suggest keep looking on our website, mostly in early next year for any potential internships for the summer of next year. I would say that as an aerospace company, we do have to follow all the ITAR regulations. So unfortunately, if somebody is an international student, then we are not able to provide them any internship. But if you have a US citizen, green card holder, then you feel free to apply when there's an opening.
Thank you so much. And any more questions? And Jim, do you have any question to Rahul?
I guess, of all of you listed a whole lot of research projects in your slides. Was there any that really stood out as really, groundbreaking or incredibly interesting and has so much potential for future in low Earth orbit or deep space?
Sure. Yeah, I do think, I mean, some of the results are still being analyzed. The one that I wanted to highlight on a separate slide, the Sweet Ride project from Axiom 4, I think that has the potential to be definitely a groundbreaking in the sense that, at least within the scope of what we did, we were mainly looking at technology maturation. But the idea is technologies like this could be useful to facilitate a wider set of individuals who could fly. So from that perspective, it has the potential in future that it could facilitate an insulin-dependent diabetic in future to we can manage their condition if somebody like that would fly. So that could be a potential groundbreaking one down the line.
Fantastic. Thank you.
Thank you so much, Rahul. And I think I would suggest the audience to go to the website link provided by Jim in the chat to look into the full presentation and look at the programs that are ongoing at Axiom Space. Thank you so much, Rahul. And let's move on to the second presentation today. Jim?
Prasad, yeah, just real quick. I don't think that next presenter, excuse me, I don't think that next presenter is here with us today. So we're going to, man, excuse me. So we'll skip the next presentation, but again, it is online. So the presentation of Helios, AI-driven sports tech countermeasure for microgravity-induced sensorimotor and neuromuscular challenges. That will be available online, both the executive summary and the full online presentation. So our next one will be Serena Pazili. Yeah.
Okay.
So go ahead
It
and
is,
introduce.
yeah. The title of the talk is Investigating the Impact of Space Flight on Human Health, Insights from the Astronauts and NUT Projects. Please welcome Serena.
Good morning, good
Presentation,
afternoon, everyone. My name is Serena Pezzilli. I am a researcher at the Human Spaceflight Office of the Italian Space Agency. And today I'm going to give you an overview of two experiments, Astronaut and NUT, that we performed during the Axiom 3 mission. Last year, in fact, the Italian Space Agency, in collaboration with the Italian Air Force, had the opportunity to participate at the third commercial mission of Access Space, a mission on 21 days on board the International Space Station. In collaboration with several national scientists, we carry out a total of 7 experiments, 5 on orbit and 2 on ground. In the next slide, I will show you the background, the study design of these two on-ground experiments, Astronaut and NUT, which aim at investigating new molecular signatures of the space environment's effect on human organisms. Even though they share the same effort to find new spaceflight biomarkers, the specific aims are a little different. In particular, astronauts try to validate circulating small non-coding RNAs as early biomarkers of the pathophysiological alteration induced by microgravity and cosmic radiation, while NUT, by comparing astronauts and submarines, try to distinguish the alteration induced by microgravity and cosmic rays, which are stressors specific to the space environment, from those induced by isolation confinement, conditions in common to the International Space Station and submarines. To reach their aims, both projects collected different biological samples from 2 astronauts of the Axiom 3 mission at different time points. One before the mission, 40 days before, and 3 after the mission. 1 day immediately after the return, 1 after 30 days, and 1 after 50 days from the return. And we collected different kind of biological fluids, which includes blood, serum, plasma, PBMC, hair, skin, saliva, urine, and several questionnaires. In the astronaut project, we collect samples to find what? Our idea is just to identify easily measurable molecules in our body by using non-invasive techniques. Molecules that are specifically modulated by space environmental stressors. With this in mind, we decide to investigate specifically different classes of small non-coding RNAs that are able to circulate in biological fluids and therefore optimal candidates as biomarkers. But how we look for them? At first, we collect samples at different time points for both astronauts of the Axiom 3 mission, and we extracted the RNAs and we sequence these RNAs by using a specific protocol for small non-coding RNAs. The sequences have been then analyzed by bioinformatic tools, and we develop a machine learning algorithm able for a longitudinal analysis along the different time points in order to identify time-modulated molecular signatures. After that, we try to validate our results in available omics database in order to increase our sample size. In the second experiment, we collected samples into different populations who live and operate in the extreme environments such as astronauts and submarines. In particular, we studied the profile of alterations induced by stress conditions in short missions, just like Axiom 3 mission, who lasted 21 days on board the International Space Station, and a similar short mission on board the Longobard submarines of 70 days. In both populations, we collected several kinds of biological fluids, including blood, hair, skin, saliva, urine questionnaires at the different time points before and after the missions. In particular, for the Axiom 3 mission, we collected samples at the same time point of astronaut project, as you can see by the figure, while for the submarines mission, we collected samples 1 day before starting the mission, immediately after the return, and 50 days after the return of the mission. And we collect samples to study what? We would like to characterize the neuroendocrine profile and finding neuroendocrine markers. We would like also to characterize metabolic profile, oxidative processes and mitochondrial function, inflammatory profile, immune function, and microbiome environment, and finally we would also to find circulating microRNAs as candidate biomarkers. In conclusion, with the astronaut projects, we would like to contribute for a more comprehensive understanding of the mechanism underlying human functional and pathological alterations in response to specific spaceflight stressors. This data will help us to develop predictive models of astronaut health risk, also identify diagnostic and therapeutic targets and novel biomarkers easily measured in situ that are able to identify early signs of astronaut stress or illness, allowing us of timely action before the development of pathologies. Moreover, if we consider astronaut as an accelerated aging model, these results will also can be translated on Earth for the identification of diagnostic markers and therapeutic targets of chronic and degenerative diseases. If you would like more information on or more details on some preliminary results of the two projects, you can find it in the longer presentation available on the Congress website. I would like to thank all the astronaut team, the Axiom 3 crew, and also the Axiom Space team. Finally, I would like to thank also the new team, the Italian Air Force, the Italian Navy, and you for the attention.
Thank you so much. This is an exciting presentation. And is Serena there?
Yes.
Yes, thank you so much. And it is really it is an exciting presentation. And uh, let me start with the question before I would like to encourage audience to go back to Q&A tool to just provide your questions. Before that, the first question is from me saying, I saw that you have used a lab on a chip qPCR technique, and in the last slide, you mentioned that that is being used. Could you please elaborate on that?
Sorry, we didn't use a cheap QCR. We use. Oh, you mean at the last slide in the node
Yeah.
project? Uh, okay. Yes. What we have done, it's really not QCR, but a sequence specific for Myrna. So it's just like similar to the first experiment astronaut, but it is only specific the analysis for Myrna, so it's not QCR.
Okay, thank you so much. There is a question from Jennifer Reed, and she asks, can you briefly mention how you have to do your model validation if your tool will
Ah.
be used to make informed decisions regarding crew health?
We don't yet done, because the problem is that at this moment we have the data only from 2 astronauts, and we try to understand how molecular signature moves in time, so before and after. And we tried also to do the same in other cores. We have downloaded some data from some study in G Labs, but the problem is that the data are very old, and we are not so able to analyze by using our protocol. So we are searching for other cores in order to validate our results. So if you have something, some data that you would like to share, I'm very glad about it.
Thank you so much. And the next question is from Elizabeth Forbes Wallace, and AST, RNA, and NUT, while both are extreme environments, would not other psychological stressors play a part? For example, those on a submarine or I assume have a mission geared toward national defense, while the ISS is for scientific research. How would the different mindset affect the results?
um, What we know is that submarines and astronauts share also the psychological stress given their missions. What we are doing now with astronauts, but with not, is just to collect information also about that by just asking to fill several questionnaires to understand the state, the several psychological outcomes, but this is ongoing. So that's it.
Thank you. Thank you so much. And the last question, I think, is from Hami Ray. She compliments your presentation, and she is asking about, she's curious about how the subjects in a submarine were used as an analog for microgravity.
No, the problem is the contrary. When we compare astronaut and submarines to try to distinguish. So in case of astronaut, you have microgravity and cosmic radiation with also isolation, hostile environment, so on. But submarines share not microgravity and cosmic ray. That means that when you compare, you find something specific for one of the two populations, you can imagine that it can be related to specific to microgravity, for example, or cosmic radiation. The problem is that you cannot distinguish the effects from microgravity and cosmic radiation because you cannot divide in the space environment. But the idea is just to use several kind of populations that can share or not some specific stressors. This is the point.
Okay. Thank you so much. And I think I see no more questions. Thank you once again, Serena, and it is an
Thank
exciting
you.
presentation. And
Thank
move on to the next presentation. And
Thank
I
you.
think
Bye.
the next presentation is resonance stimulation of the biological clock oscillation amplitude to support health and performance of astronauts in space. The talk is from Ivan Petyavev. Please, Jim, go ahead and play that video.
Good morning or good afternoon. Today I will talk about resonant stimulation of the biological clock amplitude, which could help to support the health and performance of astronauts in space. To start, a few words about biological clock hierarchy and biological clock role. The master or central biological clock is located in the suprachiasmatic nucleus of the hypothalamus. It works as a pacemaker, translating a 24-hour external light-dark cycle to internal peripheral clocks of different body tissues and organs, which help them to work together by synchronizing their own circadian rhythms. This is essential for coordination of body hormonal and metabolic reactions with its physiological activity and responses to health risk challenges. Disruption of the biological clock is one of the main health risk factors in space. So, what current countermeasures do we have to support this clock in space? Well, there are two groups. The first is physical, simulating natural 24-hour light-dark cycle with LED-changing intensity and color of the light throughout the day, using shutters or shades or wearing dark goggles, and individual noise protection, wearing earplugs. Despite implementation of these physical countermeasures, their efficacy is still insufficient. The second group is pharmaceutical, administering hypnotic medications. These can work, but, unfortunately, they have side effects that negatively affect wakefulness and performance. We propose a new approach, which can be used alongside existing countermeasures. It aims to activate the weakened biological clock in space. And we can do this by stimulating the amplitude of its oscillation. How we can do this? Let me present to you some of the lessons from our terrestrial studies. As a target for our intervention, we chose an adenosine receptors. We focused on these receptors because they control not only oscillation of the master clock in the brain, but also the biological clocks in other organs and tissues. The question is, how we can stimulate oscillation of the ADRs and activate biological clock. This graph illustrates changes in the amplitude of the circadian oscillation of adenosine receptor activity in patients with differing severity of skeletal muscle atrophy. As we can see, the more advanced the atrophy, the shorter the amplitude of the oscillation becomes. In other words, in such systemic pathology as muscle atrophy, the more it advances, the weaker the biological clock is becoming. Can we activate depressed amplitudes of the biological clock? Yes, we can by using adenosine receptor external signal, 3,5-cyclic adenosine monophosphate. This molecule is intrinsically safe. Delivered doses are within the range of its physiological concentration in the blood. Sublingual self-application, bypassing digestive tract, prevent from stomach degradation, fast delivery to the blood, essential to hit the narrow time windows for its application. This slide illustrates the principle of choosing time window. To achieve resonance in application of cyclic adenosine monophosphate, to be able to stimulate the amplitude of the biological clock. If we applied it on the descending slope of the oscillation curve, there would be no response in the activity amplitude. If we applied cyclic adenosine monophosphate on the ascending slope of the oscillation, it would push up the activity amplitude further and stimulate weakened biological clock. Could our terrestrial experience in activating biological clock be relevant and be used to address health concerns in space? What we observed in our studies is that activating the weakened biological clock can stimulate depressed hematopoiesis, which is essential for immunity, bone health, and prevention of anemia, and also stimulate depressed angiogenesis, which is essential for regrow capillaries to improve atrophic tissue vascularization hence increasing their oxygen and nutrient supply, which is critical to boost tissue regeneration. Hence, we believe that our approach could help to mitigate negative health changes in space. Our proposal is to collaborate with crew members of the International Space Station to apply resonance stimulation of their biological clock to mitigate negative changes in their health during their prolonged spaceflight missions. Thank you very much for your attention.
Thank you so much. And this is a very interesting presentation. Is Ivan there?
Ivan is in. He just needs to unmute and come on screen.
Oh, sorry, I'm here.
There he is.
Okay. There is a question in the tool, and there is a compliment from Razal. It's a great talk. Have you thought about examining circadian rhythms and their potential effects in space?
No, we haven't done any investigation in space, but we have many years of terrestrial experience to monitor circadian rhythm and trying to regulate it in our terrestrial studies.
Okay, thank you so much. And there's a next question from Paula. And what do you mean by resonance? Thank you.
Resonance is like you want to push amplitude of a swing. You need heat swing at a certain moment. If you push it swing at the wrong moment, you will not have increase in amplitude. It means you need to go a special window when a special curve of the oscillation curve. Then you can achieve increase of amplitude, like you do in swing with the children, for example, the same principle.
Thank you. Our question from Anushka Rathore, and is there a possibility for negative effects if the biological clock is stimulated for too long?
No, what we do, we're not stimulating too long. We have biological clock kind of in our bodies for all our life. We're only helping to improve amplitude or restore amplitude when it's weakened, but we cannot push beyond our physiological amplitude of the healthy, normal body.
And thank you so much. And again, the Russell is asking a question. This is not circadial, but circotidal, related to the tides, position of the moon, etc. And that is his question.
Sorry, I couldn't understand the question.
Okay, you had mentioned circadial, but he is thinking about circotidal. That is the relative to the tides, position of the moon, etc.
No, it's circadian. It's a day and light, not at the tides level.
Oh, okay. Thank you so much, and I'm looking for a few more questions, if possible. And
and I encourage, like a previous presenter suggested, go to the conference website, and there are full presentation with more details, what we actually measured, what our experience is, how this can be applied to support health in space.
Okay.
I have a question. So do you what kind of expansion of your research might you see with the commercial space stations that might be coming online?
I think if we help to support astronauts, whatever the commercial, not commercial space travels, especially when it's prolonged one, we need to support biological clock. It has been reported, it's disrupted. And whatever the mission is, we need extra support to our body. And this is not replacing anything, it's just going together with all other countermeasures.
Very good. And that applies to deep space exploration as well, obviously.
This is really, yes, because we know that the longer biological clock is disrupted, it's exacerbate pathological changes in the body. And the longer you are in a space, the more damage would be to the body. We need to support it through all the space, particularly deep missions. Yeah.
Right. Very good.
Okay. A last question from me. Have you ever planned some experiments on parabolic flights?
No, we didn't go there because I think there is not long enough to affect the biological clock. It will be mild disruption. If you go, for example, long haul flights, it will be even more disruption than a parabolic because it's longer lasting. Basically, we're not talking about impact of microgravity per se, but the length of disruption.
Okay, thank you so
And
much.
that
And
actually
go ahead.
that actually goes back to the previous presenter about submariners, submarines. Is that a logical place to do some of this testing?
Absolutely, yes. This will be
Yeah.
the place. Yeah.
Yeah. Very good.
Okay, thank you so much. And I think it's an interesting talk, and I would stress the audience to go to the website and download the presentation, full presentation, for getting more information on this particular topic. Okay, Jim, let's move on to the next talk. And that
Yeah,
is,
we'll get our presenter in. He's actually going to talk through his slides as we run them.
okay. So
So
the talk
yeah, go
is a
ahead.
self-aware AI facilitated space medical cobot.
And so Mr.
Yes,
Jafus, yeah, there you go. And so the floor is yours.
thank you. So this talk will focus is self-aware artificial intelligence facilitated space medical cobots. I'm president and CEO of Juxtopia and co-director of the Artificial Intelligence for Intelligent Augmentation think tank. Next slide, please. A little bit about me, double major of cognitive neuropsychology, computer science, Oberlin College, Howard University, master's in computer science, PhD in computer science at George Mason University, focusing on artificial intelligence embodied conversational agents. And I defended in 2005 and published in Wearable AI Enabled Augmented Reality since 2002. Served as the youngest senior software engineer at Lockheed Martin in the entire country, and direct many conferences, transition concepts to commercialization from TRL to TRL9, and secured different types of grant and contract funding, all advancing artificial intelligence and mixed reality from cases to Department of Defense. Next slide, please. This is a picture at Johns Hopkins University with some early augmented reality glasses. They were just developed as an optical see-through display and made modular so it could serve the needs for improving human performance across various industries. Next slide, please. Some of the other background Juxtopia conducted was entering a team in the Google Internet XPROD competition from 2008 to 2012. And our design here, as you see, was a modular ground vehicle that can travel 500 meters across the lunar surface with capabilities of using a near-infrared spectroscopy technique to identify water and also to come together again to perform different types of missions on our lunar planet. Next slide, please. This is another viewpoint of research that we've conducted with, at the bottom left-hand corner of the screen, you see an augmented reality view for a medic that may be in a space habitat or crew member that needs to perform a needle decompression technique as a response to a tension pneumothorax, which results oftentimes in penetrating trauma. And then on the right-hand side, we've also done significant research in artificial intelligence computer vision to identify people as well as the expressions that they exemplify or the emotions exemplified by individuals while they're in certain situations. Next slide, please. And continuing with the research and development, we have also investigated developing full body suits for space diving, for physiology within these suits, ranging from a head mounted display, as I aforementioned, with augmented reality, as well as full, immersive electromyograph, different types of spectrometers to identify non-invasively and transcutaneously biomarkers in the human, in the human being to determine early indications of injury, illness, and disease. Next slide, please. So this brings to some of the wearable computer that we developed during a NASA Marshall Space Flight Center contract with Tuskegee University, where human beings need to perform complex tasks, ranging from doing repairs in a space habitat, or providing clinical expertise, or performing a clinical task during long duration space exploration. Here, this is called the Juxtopia Imhotep band on the right-hand side. I'll talk about that first. And then the augmented reality glasses on the left. And on the Juxtopia Imhotep band, it includes an inertial measurement unit, an electromyograph sensor, as well as battery, in order to track continuously psychomotor movements of the crew's hands or arm while performing a task, and then running long short-term memory neural network as a time series type of AI model that is built from the data that's collected in the data set after being pre-processed and segmented, so that each step along in the task can be evaluated correct or incorrect, and then provide that real-time feedback through speech synthesis, another form of artificial intelligence, to the crew member. On the left-hand side is the ruggedized mill standard 810H design augmented reality goggle. We were originally funded by DoD Rapid Innovation Fund from TRL6 to TRL9. And then we have also secured cases funding. And now we've modularized a more ruggedized frame for integration of an optical see-through display, plug and play. Next slide, please. Here with the research, what we've investigated is cobot. So given an augmented reality display, given the armlet that I just mentioned, then together the augmented reality display is worn by a crew member. The crew member here is depicted in a controlled study by the young man in the two bottom pictures, performing a task on a pallet. A pallet most likely will be integrated in the space habitat. So donned with augmented reality glasses, this individual, simulating a crew member, can perform a task, step through the instructions by speaking to the artificial intelligence assistant using speech recognition, speech synthesis, and then the data is collected from the armbands, so that if any procedure looks like it's being performed incorrectly, because the data then is being compared to a already trained AI model, then the assistant can make corrective action on the fly. Also, because a large behavioral model is generated, then the autonomous robot, which you see on the top left, if it's a clinical application, can then be applied to collaborate with a human being, perform certain complementary tasks with the crew member, and then make sure that sustainability of life is preserved and mistakes are mitigated. Next slide, please. I'll quickly go through these slides. So in the top left-hand corner of the slide, this is background research on what we've done with comic medics on the battlefield before they go and actually serve. They are trained at Fort Sam Houston if they're 18 Delta or 68 Whiskies. So we've done significant research with wearable augmented reality for psychomotor proficiency and competency, clinical skills ranging from needle decompression, chest tube, cricothyrotomy, pressure bandage, tourniquet, IV. On the right-hand side, you see an EMT administering a tourniquet, and same intervention, augmented reality goggles, two different types of head-mounted displays running the same software. Next slide.
Next.
And you'll see here, well, we can quickly proceed through the slides, but this medic right here is performing a cricothyrotomy and using speech recognition to perform the task. Next view. You can see here the EMT performing the same type of, different type of procedure, but stepping through the augmented reality instruction with speech and getting assistance on demand. Next slide. And in the space habitats for long duration space exploration and the Artemis mission, crew will be required to be in close quarters for long periods of time without, having obviating the need for mission command and control in practice for planets like Mars and other exoplanets in the future, and consequently what we do now in order to provide crew with on-demand assistance using wearable computers and artificial intelligence is paramount. Next slide. So the team members, we have an array of team members, colleges, universities on the left-hand side, as well as the wonderful team of Juxtopia, the Juxtopia engineering team. And we are continuously developing and commercializing products that you saw to make sure that we improve so we're continuously improving the performance of crew for long duration space exploration. Thank you much.
Okay, thank you so much. And I think it was a very fascinating talk. And let me lead a question. What was you talked about the long terrain vehicle on the lunar surface, and can you talk about its size, length, and what was its performance?
Yes, so that's a great question. Our performance metrics are based on dependent variables of time, accuracy, and completeness. So we did an institutional review board study before, and also qualitative research. So we looked at time, accuracy, and completeness when an individual is wearing augmented reality performing a skill. What we found out unintuitively is that when a person is wearing the augmented reality goggles and performing a task where the individual is a novice performing that task, the time is increased in that task. So there more time is allocated to perform that task. When that crew member or that person is performing more time on that task. They told us that the individual is paying attention to every step that's holographically projected in the augmented reality display. Conversely, the errors are significantly decreased because now the individual is the crew member or simulated crew is going through each step, taking the time, looking at the instructions, decreasing the errors. Control group, less time to perform the task, but increased in errors. So we see that inverse relationship. In both groups, the tasks were completed.
Okay, thank you so much. And I think in the last one of the last slides, you mentioned about, or you showed a picture of habitat. Has it been constructed, or is it like inflatable or a fixed? Or can you talk about that habitat structure?
Yes. So we're not responsible or doing habitat construction or architecture. But from what I know, the habitats that are being designed right now are for around about three-member crew, almost like a bunk bed style, with your work environment in that habitat, as well as your exercise and your personal space. So very small quarters for 3 people for very long periods of time. I'm anticipating like 6 to 8 months at a time.
Thank you so much. And one more question. Have you done any augmented reality experiments on ISS?
We have not done ISS experiments, augmented reality experiments on ISS. In 2015, we did secure a Center for the Advancement of Science in Space, and we were funded to do a mouse models, biotechnology type of research with our augmented reality with a glove box. But we did not proceed with flying equipment on the International Space Station, but we did develop a lot of ground testing for that because mice are expensive, or rats are expensive on the International Space Station. So we had to do ground studies instead.
Thank you so much. And a question from Elizabeth regarding the armband. Are there any differences noticed regarding execution of tasks by males versus females? And also, could there be more male data processed by AI so that when a woman uses it might indicate an error due to psychomotor differences?
And if I understand the question correctly, are there any significant differences between gender, male or female, performing a psychomotor task? Is that correct?
That's correct.
We have not done that research, but in our studies, institutional review board study, we have a even distribution of women and men who participate in our groups. We have not seen any differences between what the inertial measurement unit, so you have a linear acceleration, angular velocity, absolute orientation, and goes into a complete data set, XYZ, contiguously. So linear acceleration, XYZ, absolute orientation, XYZ, absolute orientation, XYZ, and as many EMG, electromyograph data points, which are floating points that are populated in the data set. And we don't see much of the differences in the IMU certain, but we can potentially see, depending on the muscle tissue between genders, male and female specifically, there may be some differences in muscle activation that we could look at in the future. As of yet, we haven't done that research, but that's a great recommendation that we could look at that in the future.
Thank you so much. And a question from Christina Johnson. This augmentation is absolutely fascinating. Thank you for sharing about your work with us. Do you see this tech as being helpful with attending to science experiments and maintaining aeroponic systems for crop production?
Absolutely. So as you probably know, crop production in a space habitat, growing food for crew, is an approved mission as in Artemis, from my understanding. And I can definitely see, yes, so if crew have not done this before, they need assistance, then they put on the glasses, the augmented reality, the Juxtopia augmented reality glasses, speech request, the steps to cultivate crop on board the space habitat, know when to water, know when to add other nutrients in the crop, and get the timing of that right there in the head mounted display. If there is a specific technique of putting a camera, and that's just one of our simulations, to rig a camera and camera on a pallet, and it has to be connected to a mill standard, defined outlet. That's not like the current outlet that we have on Earth in different countries, it's different. Then that connection point must be done very well without mistakes. So that can also be something that can be presented on the augmented reality display, reducing errors. The armbands, again, because it's tracking the orientation and acceleration of the arm over time, then there's an assistant, we call that assistant Jizzy, Juxtopia Intelligent Space Instructor, can then continuously provide autonomous feedback. Command and control is not going to be there when we go to Mars, so it's going to be very important in the future.
Thank you so much, Jeffers. And again, a question from Manjula. Is it 3D printed? I'm not sure, but again, is it 3D printed?
So our initial prototypes are 3D printed to prevent doing injection molding, which is a very expensive process, as you know. So our early prototypes, they're 3D printed with different material, ABS plastic to nylon. Yes.
Okay, thank you. And Ajay Kumar asks, he needs your contact, and I think in your long presentation, your contact is there for people to contact you.
Yes, and if it's not there, if I miss putting there, then the best way of reaching me is on my email address. It is japheth@juxtopia.com. And for everyone here, I will also put that in the chat for you to contact me.
Thank you so much. Japheth. And yeah. So a question from George Frias. Fascinating research and excellent presentation. Have you done any investigations in microgravity at all, aside from ISS?
That's another study we would like to do, microgravity test. We haven't done any microgravity testing of this thus far. We're looking for partners to work with us so we can do some microgravity testing.
Oh, thank you so much. And I think there is one more question. Lisa say, thank you, Dr. Doswell, for your presentation. And will regolith materials be used for prototyping the lunar habitats?
Can you repeat the question, please?
Yeah, will regolith materials be used for prototyping the lunar habitats?
I'm not too familiar with how the lunar habitat in itself would be produced. I know Marshall Space Flight Center is working with other industry partners for the space habitat itself. I can say for the material of the head-mounted display, our augmented reality and our wearables, then they would have to be, after the 3D printing, we do this for form factor, but materials that can be sustained in, microgravity environments, even for long durations of manufacturing on planet. So we've looked at in situ manufacturing. What materials are on planet that can be used to manufacture the wearables, from the wearable augmented reality chassis to the actual optical lens to the electrical components as well, doing something that's a little bit more transformational and revolutionary than a printed circuit board integrating transistors and on a, or different components on a PCB. So we're looking at these types of materials for long-term sustainability and manufacturing on planet.
So thank you so much. And once again, it's an excellent presentation, as well as your answers were very inspiring as well as interesting. And I would like to let the audience know that they can go to the website, ISSRDC 2025 website, and download his presentation with a lot of information there. And now, thank you so much, Jeffers. And we are now we're now coming to the last presentation of the session, which is Acquire's antimicrobial biofilm inhibitors for environmental control and life support. The talk is from Marilyn Bruno. Jim, could you please play that?
Greetings from San Diego. I'm Marilyn Bruno, CEO and co-founder of Acquire, to tell you about our bio-inspired chemicals that clean water and surfaces from deep sea to deep space. Imagine our excitement when NASA contacted us to solve a problem that they were encountering in the water reuse recycling system used on board the International Space Station. And there it is. The problem is biofilm. This glue-like substance seen in the upper left is from a photo that NASA sent us of biofilm clogging the Environmental Control Life Support System, called ECLAS, that astronauts rely on to take their urine, soapy water, and humidity from the air to recycle it through a complex filtration system into potable water every day. Because Acquire's co-founder, Dr. Cynthia Brazel, is a marine and medical microbiologist specialized in biofilm, she knew that we could solve the problem. Biofilm is formed when two or more bacteria or fungi jointly work to colonize a surface. The biofilm is thicker and stickier for surfaces in contact with water. At that point, the underlying bacteria or fungi are 100 times harder to remove from the surface. The bacteria or fungi then envelop themselves in biofilm that grows thicker and faster in direct proportion to their hazards in their environment, such as antimicrobials, physical removal, biocides, scraping, sterilization, UV light, sonication, antibiotics, and the human immune system. At that point, the underlying bacteria and fungi are 1,000 times harder to kill and called superbugs. Nothing can kill them. In space, biofilm forms even thicker and faster than on Earth due to radiation, the vacuum, microgravity, etc., technically making all bacteria and fungi in space superbugs. Sure enough, astronauts drinking water from the contaminated E-Class reuse recycling system all became ill and had to be sent back to Earth. So how did the microorganisms get on board the ISS, which was manufactured in a clean room and using silver, a natural antimicrobial? The answer is that they hitchhiked up there, given all the environmental threats, quickly developed resistance. They even developed resistance to silver. And there was the remedy all along. Cynthia Bursell saw this very scene, a healthy coral reef with clean surfaces without fouling, and hypothesized that nature had developed the antidote to biofilm formation. She collected materials from the clean reef surfaces and, upon analysis, discovered a new genus and several new species of marine microbes that produce hundreds of chemicals that work at low doses to simply prevent the unwanted bacteria and fungi from attaching. Since they can't attach, they can't form biofilm, colonize, contaminate, foul, or infect. ACOR initiated a 3-year project with NASA's Marshall Space Flight Center to test the efficacy of its chemicals. Here's the proof of these claims. After dropping our product into the E-Class system simulator, 99.99% of all contamination was removed in minutes. In fact, 1 dose of ACOR's treatment eliminated biofilm from the E-Class system for 15 months without replenishment. Bottom line, ACOR solved NASA's problem and was awarded NASA's iTech Award in the life support category. As NASA Health stated, without ACOR, nobody would arrive alive on Mars. Of course, the astronauts thank us for clean, recycled drinking water. But the biofilm problem is not going away. In 2018, NASA's Jet Propulsion Lab published a paper saying that they had identified over 130 biofilm-forming pathogens on surfaces inside the ISS. Today, that number is higher as we learned that the disinfectant wipes on surfaces, etc., were no longer effective. And in 2022, our product was launched up to the ISS for further testing. In conclusion, ACOR is ready to solve problems and current and future challenges to space travel and exploration, particularly as astronauts rely on clean instrumentation, water, and environments for life support, growing crops, health, and hygiene. We welcome collaborations and look forward to hearing from you.
Thank you so much, and Marilyn. And there's a question from the audience from Russell Higby. While silver is a recognized antimicrobial, have you considered low-dose iodine?
Yes, well, working with Marshall Space Flight Center, we learned that silver was the antimicrobial of choice because there's no way for hazardous chemicals, which require hazmat protocols for use, storage, and disposal. There's no way to dispose of them. So having a natural, non-toxic, effective remedy is ideal. They're continuing to use silver, of course, but the bacteria develop resistance so fast. It was frankly astonishing.
Thank you. Joe Adam asks, is a built environment probiotic dosing the ISS with known healthy environmental microbes feasible?
Well, I'm not sure how to answer that. If you send me an email, I'll have our Cynthia Bursell, who's the microbiologist, answer that question. I'm not qualified. But technically, in all of our work with DOD, Department of Energy, Department of Agriculture, EPA, our product works against a broad spectrum of bacteria and fungi. We have done surface testing. I'm not sure if your question is regarding the good bacteria. Does it affect them? I cannot tell you. We would have to do case-specific testing.
Thank you. Sabhi Anzak asks, has data been collected on acquire biocompatibility for microorganism or tissue culture? And what's the procedure for using this for experiments?
Yes, we've discovered chemicals in the ocean. We've have natural chemicals in milligram to gram quantities. We've synthesized the most active ones to over 70 analogs are synthesized and patented. We've not done tests in human tissues. We've done surfaces only. And we look forward to partnering with companies that have the capability of doing further testing. You know that antimicrobial resistance is already killing 2 million people on Earth because there are no cures for these pathogens. They're resistant to everything. And we have the opportunity to do further experimentation, but we're not chemists and we're not pharma chemists. So we look for partners at this point. We feel that ACOR has done the discovery, and now the next step in our development is to partner to see if we can incorporate these chemicals and coatings and materials to avoid contamination from the get-go, and certainly in human health, animal health, and plant health, because biofilm is literally everywhere, and especially in environments where there's water. Curiously, you may ask, is our product currently being used on board the ISS? The answer is no. NASA is shipping up water with every launch. They're not risking the E-class system. Okay. And so we are seeing that that's great for short duration missions, or where missions can be supplemented, but not for the long duration manned space travel.
Okay, thank you so much. And the question from Chris Brunkhorst: is there any risk of ACOR negatively impacting the astronauts' gut microbiome?
Yes, that's the issue. We've not tested inside the body because of that very issue. We are looking at topical infections and surface degradation and clogging and biofilm formation. Biofilm originates corrosion, scale, and contamination on surfaces. So we are interested in knowing if contamination is impacting instrumentation as well.
Thank you so much. And Russell Higby has a follow-on question. Iodine is a natural antimicrobial that we eat daily. Hopefully, BZK, benzalkonium chloride, is considered the standard wipe on station. And can you speak to how effectively it is or it isn't?
All I can do is anecdotally comment with our discussions with Jet Propulsion Lab, which identified the 130 biofilm forming bacteria on board surfaces inside the cabin. And the fact that they were there indicates that the wipes are not working. So the fact that they are reproducing indicates the wipes are not eliminating them. So the interest is to eliminate them. I might also mention, you saw that picture of actual biofilm inside the component of the E-class system. In microgravity, it's growing like a Dr. Seuss structure. It's weird. It's not like anything found on Earth. It's thick. It's really hideous. And if they tried to remove it with every known antimicrobial and it hasn't worked, that's why they called us. So we're happy to contribute, but as I say, we're a small company looking for partnerships and hope that this can lead to that. Thank you so much, Jim, for the opportunity, and for Nashim for this session. Thank you.
And also, I think 2 more questions or just a comment.
Oh, excellent.
Sorry, the anonymous attendee just compliments your presentations and congratulates your work. And the question is, has this technology being patented?
Oh, yes. We have many patents on 70 chemicals. 30 are novel, completely novel, synthesized chemicals. The actual natural chemicals are our trade secrets. The synthesized analogs, we have 40 synthesized analogs that are EPA approved for our phase 1 rollout, which are water treatments and surface cleaners. And that we are handling in-house pretty well with different water treatment, water reuse, recycling, improving filtration so that PFAS can be better absorbed. We're using it in algae cultivation with DOE for the last 5 years, very deeply involved in algae and hydroponics. We have done a lot of literally 7 years of validation testing at pilot field trials, et cetera. So we're very, very confident in what we're launching works. So we're starting with phase 1, which are water treatments and surface cleaners, and look forward to the rest of the phases that ACOR can be used for. But as I say, that's what we've got so far. We're on the manifest for another flight to the ISS. They're very excited about that.
Thank you so much. And I think Russell Higby again comes back, and congratulations, compliments your presentation being very inspiring. So
Thank you
thank you.
so much.
Thank you, Jim. Take it away.
All right. Yes, thank you, Dr. Bruno. Fascinating stuff. And thank you to everyone for being here for this session. We're going to take a short break now, and we'll get started with our next one that starts at 11:50. So in 15 minutes or a little less than that, we'll come back for fundamental biology. We're going to leave the webinar running for that time. So just hang tight, and we'll be back in a few. Thanks all.